So first of all, I'd like to thank the opportunity from the conference allowing me to present my work here. My name is Mengping Liu from the University of Hong Kong, and my supervisor is Professor Julian Tanner. And today's presentation is about the aptamer evolution against the BMP2 and the application of this aptamer to develop an extracellular matrix mimetic scaffolds for BMP2 delivery. The presentation will cover basically 4 parts of work. And first of all, I will start off by a brief introduction of the research background. And then I will move on to the aptamer selection, and followed by is the development of the aptamer functionalized BMP2 scaffolds. Yeah, we end up this presentation with a brief conclusion. Well, this project meaning about BMP2. BMP2 is a growth factor, and that's approved by the US FDA in 2002 for bone regeneration applications. Clinically, BMP2 is injected into its scaffold, which is an absorbable collagen sponge. But since the collagen sponge cannot capture BMP2 specifically and tightly, and that could lead to burst drug release and several physiological deaths of BMP2 local sites. These issues have been found and cause various side effects in patients. For example, the ectopic bone formation, post-operative inflammation, and bone resorption. So that creates a research gap in this area, which is scaffolds allowing specific drug capture and sustainable drug release are not available for BMP2. That drew me to design an alternative scaffold for BMP2. And the design of the scaffold in this study was kind of inspired by the natural extracellular matrix. This is a bioenvironment. Yeah, the image of this matrix represented here, and it could regulate cell adhesion, proliferation, and further induce the production of new bonds. Inside, type 1 collagen fibers and growth factors actually are the key components. They could provide mechanical support and stimulate the osteoinductive syndrome pathways for bone regeneration. And based on that, we designed scaffold, and that's assembled with the collagen fibers and the DNA aptamers specific to BMP2 to mimic the function and the function of a natural extracellular matrix. To achieve that, 2 objectives are proposed. The first of 4 is to involve BMP2 specific DNA aptamers. Second is to apply these aptamers to develop aptamer functionalized scaffolds for BMP2. And then, let's see the work about aptamer selection first. This is we use very classical bit-based strategy to perform the aptamer selection. Because our targets BMP2 were expressed in E. coli with Histax. This is a purified BMP2 dimer. It's very easy to conjugate and that into the nickel magnetic base. And after 22 rounds of selections, we were able to evolve a DNA pool that demonstrated high binding affinity and specificity to the target. And then that was sent for sequencing, and several representative DNA sequences were selected for characterization. Of this, we found that BA1, this DNA sequence actually was very good aptamer candidate because it showed very strong binding affinity and specificity to the target BMP2. Next, we performed molecular simulation studies and to investigate the putative interaction models of the aptamer with BMP2. The figure on the right, on the left is the interaction models we predicted between BA1 and BMP2. On the right is the one formed with Guillain-Barré sequence. Right, several parameters about these 2 models were listed in the table. And algae score and max up this set of data basically suggests that these 2 models reached a good standard. And free energy values suggest that the interaction models between BA1 and BMP2 was much more stable than one formed with Guillain-Barré sequence. And one more interesting observation here is that almost 50% of the binding nucleotides in BA1 was most likely to bind to the heparin binding site of BMP2. To verify that, and then we performed competitive binding assays. In this assay, heparin was used as a binding competitor to BA1, and we designed 3 binding models. And the first model, BA1-heparin, means BA1 was added first, and then followed by the heparin when incubating with BMP2. The second model just reversed the first one. The third model is to add heparin and BA1 together when incubating with BMP2. All right, and then EMSA and Ilona experiments were performed to evaluate the binding behavior of BA1 under different scenarios. And basically, this data suggests that heparin and BA1, they indeed have competitive relationships when binding to BMP2. And heparin was able to block the binding of BA1 to BMP2. And this kind of results was further confirmed with a heparin-like molecule, which is a polyphosphate. So this set of results basically indicate that BA1 and heparin likely to share same binding domain, which is the heparin binding domain when recognizing BMP2. In summary, this part of work, we were able to evolve DNA aptamer to BMP2, and this aptamer exhibited high binding affinity and specificity to the target. And also the molecular simulation study suggests that BA1 was likely to mainly target the heparin binding site or BMP2. And next, and then we applied this aptamer to fabricate the scaffold for BMP2, right? And we took use of DNA conjugate self-assembly technology to construct this fibrous scaffold. Yeah, the image here shows the morphologies of the fiber is actually constructed using different formulations. And by seeing this imagery, we can see that with lower current diffractions in the formulations, the fiber were shorter and thinner. By increasing the current diffractions, and the fibers become more aggregated and elongated. But there is no much difference between the fibers formed with Grammar sequence and the BA1 aptamers. A further characterization of this fiber is, was presented here. And 2 elements in the fiber is current DNA were labeled using different fluorophores. And we were able to observe the co-localization of these 2 elements in the fibers, just confirming the fibers were indeed assembled with DNA and current molecules. Next, and then we evaluated the binding affinity of the aptamers on the fibers to BMP2. And it's found that the BA1 aptamers could maintain its high binding affinity and the specificity to BMP2, particularly when it's assembled in the fibers with 10% of coverage. And that was much higher than free BA1. And possible reason we guess that because the fibers allow a higher density of the BA1 to be displayed at the scaffold surface. But by increasing the current diffractions, we can see a decrease of the binding affinity of the aptamer. And one possible reason we guess might be because this change actually lead to the reduction of the surface area to volume ratio, and that could further reduce the loading capacity of these materials to the aptamer. And next, and then we applied these, and then we evaluated the impacts of the fibers, particularly the one assembled with 10% coverage on the BMP2-induced osteogenesis effects. The first osteoinductive factor of BMP2 we look into is the alkaline phosphatase expression level. BMP2 was able to induce the production of alkaline phosphatase in the C2C12 cells. And once loaded on the aptamer functional scaffold, we can see a significant increase of the alkaline phosphatase expression level. And this improvements was very specific and when compared to the fibers assembled with scrambled sequence. And the second osteoinductive factors we investigated is the cell adhesion behavior. After BMP2 treatment, the cells, we can see they become more elongated and stretched, indicating the occurrence of the cell adhesion. And when it's delivered by the aptamer functional scaffold, we can observe the similar phenomenon. Furthermore, the cell number was markedly increased also, when compared to the fibers with the scramble sequence, these improvements definitely was much more, it was very specific. In the end, we performed wood healing assay. Just use this experiment to mimic the process of bone regeneration in cells. And the data showed that BMP2 treatment could promote the wood healing process. And furthermore, the aptamer function as a scaffold could just accelerate and the BMP2 induce the wood healing process. And this trend was found very specific when compared to the scramble sequence assembled fibers. In summary, in this hard work, we used the BA1 aptamer to assemble fibrous scaffold for BMP2, and this scaffold was found to demonstrate effects and to promote BMP2 induced osteogenesis. In conclusion, so first of all, we involved BMP2 binding aptamers, and then we applied this aptamer to function as a collagen fiber scaffold that could promote BMP2 induced osteogenesis, and indicating this scaffold may have a potential for bone regeneration application. But before that, lots of work need to be done. For example, the aptamers in the fibers should be further stabilized, and the putative interaction models should be resolved, like for example, cryo-EM, and the effects on the bone regeneration of these fibers can be validated by appropriate animal models. That's all for the presentation, and I also like to take this opportunity to express my gratitude to my supervisor, Professor Julian Tanner, and all of my lab mates in JET Lab. Thank you. Welcome questions.